Hello everybody. Today in this video, we'll study about oogenesis. So this is the part second video of the gametogenesis. In the first, we have studied about the gametogenesis and spermatogenesis. You can watch the previous video in the Medico channel, that is Medicos 1.0. So in this video, we'll precisely talk only about the oogenesis. So at first, what is oogenesis? The process of formation of ova from the primordial germ cell involving the cell division, mitosis and meiosis is known as oogenesis. So when does the oogenesis begins? Begins within the uterus of the mother during the intrauterine life. So in case of female, the oogenesis begin already in the uterus of the mother during the intrauterine life. This separate oogenesis from the spermatogenesis, where the spermatogenesis occurs only after puberty in case of male. So what are the different processes involving the oogenesis? At first, the primordial germ cell, 2n, 2n represent the diploid structure, develops into oogonia. Majority of oogonia continue to divide by mitosis to form primary oocytes. All the primary oocytes are formed by month 5 of the fetal life. That is, at first, the primordial germ cells. So, in order to understand what is primordial germ cell, you can watch my previous video in which I have already explained about the primordial germ cell. At first, the primordial germ cell divide by mitosis to form the primary oocytes. The primary oocytes, all the primary oocytes are formed by month 5 of fetal life, which means that no further formation of primary oocyte occurs after month 5 of the fetal life in case of females. Now, this formed primary oocyte enter the prophase first of meiosis first, but at diplotin stage, it get arrested due to oocyte maturation inhibitor or OMI, which alters the level of cyclic AMP. That means that the primary oocyte undergo development, but at prophase first of meiosis first. At diplotin stage, it get arrested due to oocyte maturation inhibitor, which alters the level of cyclic adenosine monophosphate. Now, this primary oocyte remain arrested at prophase first until and unless the LS surge occurs, which occurs only after puberty. So, in case of female, the luteinizing hormone surge occurs after the menarche or when the menstruation starts. So, after LS surge, a primary oocyte completes meiosis first to form two daughter cells. So, out of two daughter cells, the first one is the secondary oocyte. The primary oocyte is diploid due to meiosis first. The secondary oocyte is a haploid structure, and the second one is the first polar body, which later degenerates. Now, this formed secondary oocyte also promptly begin meiosis second, but is again arrested in the metaphase of the meiosis second about 3 hours before ovulation. So, the secondary oocyte, as soon as it is formed, it undergo development by meiosis second, which is similar to that of mitosis, but is arrested at metaphase stage 3 hours before ovulation. So, the two condition arises. The secondary oocyte completes its meiosis second only if the fertilization occurs. So, same thing written here. If the fertilization occurs, secondary oocyte completes meiosis second to form mature oocytes. But what happens if the fertilization does not occur? If the fertilization does not occur, the secondary oocytes degenerates after 24 hours of ovulation. So, this is all about the oogenesis. Now, we have few special points. Approximate number of primary oocyte at fifth month of intrauterine life is 7 million. So, on further development, it undergoes degenerating. So, the number of primary oocyte decline to 2 million at birth, 40,000 at puberty, And out of 40,000, only 400 to 500 undergo ovulation in the female reproductive life. So, this decrease in the oocyte is due to degeneration. This is all about the oogenesis in this video. We can better understand in the picture. This is the oogonia having 46 chromosome, which is the diploid, and 2n DNA. Content of DNA is 2n. Due to DNA duplication, the primary oocytes are formed, having the diploid chromosome number 46, but the DNA content is doubled, that is 4n. Now, this primary oocyte is arrested and dormant until puberty, arrested at diploidine stage of meiosis second. So, due to LS surge in puberty, the primary oocyte undergo meiosis first cell division to form the secondary oocyte, which is haploid 23 chromosome, and the first polar body, which degenerates. Now, the secondary oocyte is further arrested in the metaphase of meiosis second, and only if the fertilization occurs, it completes meiosis second to give rise to mature oocytes, which is haploid, and the second polar body. So two polar bodies are formed, first polar body and the second polar body, which later degenerates. So, from the one primary oocyte, one mature oocyte is only formed, and the two polar bodies are formed. So, this is the figure showing oogenesis. This is all about the oogenesis in this video. Thank you for watching. For more informative videos, please subscribe Medicos 1.0. Thank you, everybody.